Good evening, everyone. I'd like to welcome you to the first of the keynote addresses. And I'll have to say that it is both a privilege and a pleasure to welcome David Baltimore as this evening's speaker. And he's had such a fabulous and productive career. I think there are lessons for all of us. The first of which I think is that he started early. And everyone should look and see if they can pick David out. And if you can't, that's David at the Jackson Labs in the summer of 1955 when he was 17. And in one of life's interesting coincidences, that gentleman at the same time, same place, is Howard Temin. David has a long and distinguished connection to Cold Spring Harbor. He was in what I think was the first of the undergraduate summer research programs, and this is David at 21 at Cold Spring Harbor in the undergraduate research program. And of course, it was only really 11 short years later that he wrote this truly seminal paper that was published back to back with a related paper from Howard Temin. And I think this is what David's going to talk about tonight, so I don't want to tell you or try and tell you anything he'll tell you better. But there are two things about this paper that I think are worth pointing out this evening. One is this is a single author paper. And I think he'll probably show you that the experiments in this paper were experiments David did with his own hands. The other thing that I think is notable today is that this work was supported by grants from the U.S. Public Health Service and the American Cancer Society. And in a time when there have been questions about whether or not it's a good idea for the United States to continue to support basic research, this is exactly the kind of thing that I think is clear proof of the value of that kind of research. I'll ask for a show of hands now. Is there anybody in this room who has not used reverse transcriptase in their research? And if I didn't expect to see any hands, and I don't, but if I had, I would have said, well, if you haven't, you will. And the important idea here is that not only is this an incredibly important scientific discovery that revolutionized how we thought about many problems, it had enormous impact beyond the research laboratory. It's been critical in the clinic, involved in various types of diagnostics, and the entire biotechnology industry rests on the ability to use RT. And you could imagine, I think, if you were someone who had done Nobel Prize winning work at age 32 and won the prize at 37, that you might have said, well, you know, take it easy a little bit. And that's not at all the way it worked. Before actually RT, there was the discovery with Alice Huang of the VSV RNA polymerase, which is found in virions. And some of the work on RNA polymerases and virions prefigures the work on reverse transcriptase. There was some very important recognition of the fact that defective interfering particles were really important in a whole variety of viral infections. Then there was RT. And after RT, there were a whole set of lovely contributions to our understanding of retroviral replication. There were development of some of the early murine retroviral vectors. And of course, in those days, it was the RNA tumor viruses. And so there was work on oncogenes. And this is not in any way intended to be the limit of it. These are scientific highlights, and I'm being very selective. So there was work on the Abelson gene in the context of the Abelson virus. But that, of course, led later to the recognition that in human tumors, the BCR-ABL fusion was, in fact, a driver of human cancer. There was, of course, the discovery of NF-kappa B, and following later, the discovery of the important regulator and inhibitor I-kappa B, RAG1 and RAG2 involved in the generation of antibody diversity, microRNAs even more recently, and the thing that's really sort of scary about this is highlights. And it's selected. I could have chosen completely different things. And again, you might think that someone who was that successful in the laboratory might not be doing things outside the laboratory, and you'd been wrong about that too. He was one of the organizers of the Asilomar conference that gave us in this country what I think was a reasonably rational approach to regulating in a safe way the original work with recombinant DNA. He was the founder and first director of the Whitehead, president of Rockefeller, and of course, president of Caltech. And lest you think that this sort of two careers, both in terms of the overall management of scientific enterprises, universities, and the science in his own lab was sufficient, I want to say that last but not least, David is a fly fisherman. I give you David Baltimore.
Thank you so much. Am I being heard? I have no idea. Tell me, am I being heard?
Yes.
We hear you loud and clear, David.
Oh, okay, fine. I just don't get any feedback. Thank you so much. That was a wonderful introduction. In fact, I feel like I should just take a bow and bow out. But I'm going to give you an idea of the history of virology from my own personal point of view. So I've been studying viruses, as was said, now for over 60 years. I started in 1961 as a graduate student at MIT, and I started in virology for the same reason that the phage workers started working with viruses, which was to learn about genes. And the genes that we wanted to learn about were the genes of the viruses. The pioneers in virology built on the foundation which was established by a French-Canadian scientist named D'Herelle, who discovered bacteriophage and who in 1917 showed that the virus could form plaques on a lawn of bacteria. And these are the plaques of lambda bacteriophage. And that allowed us to count. And counting meant that you could do quantitative work. And so it opened up quantitative virology. Now, in the 1930s, Max Delbruck, who was a physicist working in Germany, was intrigued about whether there was a way of studying viruses for their genes. And he picked up the work that D'Herelle had done, and he brought it to the United States in 1939. Now, 1939 was the beginning of the Great War in Europe. And that war chased out of Europe the beginning work in virology. When Max took it to Caltech and worked with a man named Emory Ellis, who was already at Caltech, and they showed that phage replication involved a one-step burst of virus from infected cells. And really, that opened up the ability to study the life cycle of viruses. Delbruck stayed in the United States at Vanderbilt University and met with another European, Salvador Luria, who had been chased out of Italy, and Al Hershey, who joined them in 1943, an American. And together they established the phage group. And the phage group met in summers at Cold Spring Harbor. Delbruck went back to Caltech and spent the rest of his career there. Luria went to various universities in the heartland of America and ultimately came to MIT. And these two engineering schools, Caltech and MIT, became the powerhouses of molecular biology because of those two immigrants to the United States. And here they are in a famous picture taken at Cold Spring Harbor in the 1940s. So in my own personal life, I met Luria at Cold Spring Harbor just as he was coming to MIT. I was then a junior at Swarthmore College doing a summer, having a summer opportunity at Cold Spring Harbor. And Luria invited me to be a graduate student at MIT, where he was setting up what was one of the very first programs in molecular biology in the United States. And I started working with phage, in fact, with Cyrus Leventhal. And one day I said to Luria, might it be possible to work on viruses that infect animal cells rather than viruses that infect bacteria? And Luria said he had no idea whether animal viruses could be studied in that way. But he set me up a discovery plan. And that was to work with a quantitative animal virologist, Philip Marcus, at Albert Einstein College of Medicine for half of the next summer. And then for the second half, to take the Cold Spring Harbor course in animal virology, which had just been started and was taught by Richard Franklin along with Ed Simon. And I saw that the field was ripe for discovery. It was just sitting there waiting to be picked. And so I moved to the Rockefeller University, where Franklin was a faculty member, and did my thesis with him, and was able to do quantitative work because of the work of Delbecco and Vogt, who had shown that you could make plaques of animal viruses just as you could make plaques of bacteriophage on monolayers of animal cells. So I did my thesis in what was effectively 2 years of research and worked on RNA-containing viruses because that's what Richard Franklin worked on. In particular on poliovirus, relatives of poliovirus. And it was literally a discovery of a week, per week. I published more than 15 papers during my graduate work. And the key question that I was working on was, how can RNA be replicated? And there were two possibilities at that time. One was that the virus encoded a replicase of some sort. The other was that cells could replicate RNA because we had no idea whether they might or might not have an RNA replicase. And I actually spent some time trying to find an RNA replicase in animal cells and could never find it, but negative results are not totally satisfying. And in 1962, I found the style of experimentation that I did for the rest of my life, actually, which was to look for enzymes that carry out key events in the life cycle of a virus, in particular that replicate RNA. And what I found was doing biochemistry now, really taking a page from the work of Arthur Kornberg. And very quickly I showed that infected cells had a polymerization enzyme, and that was the mangovirus RNA polymerase, the first enzyme ever found to copy a template RNA into a product RNA. Oh, wait a minute. So RNA to RNA replication remained the paradigm of RNA virus strategy for the next 8 years. And it kept us busy working first actually in California and then at MIT. And as was just described in the introduction, one of the things that we did was to decide after 8 years to move away from poliovirus and to look at other viruses, particularly lipid-enclosed viruses, and rapidly showed that vesicular stomatitis virus, which was a somewhat obscure virus, worked with by my wife, Alice Wong, who was at that time a postdoctoral fellow with me. And she said, maybe we should find out how these viruses replicate. So we asked an incoming student to work on vesicular stomatitis virus, and she found very quickly that the messenger RNAs for this virus in infected cells were not the virion RNA as it was for poliovirus, but rather were subviral short messenger RNAs. And when we hybridized those messenger RNAs to virion RNA, they hybridized. And wait a minute now. And so they were negative strands in the virion. And that presented a very puzzling problem of how does an infection get started? And we showed that, in fact, there was a polymerase in the virus particles that copied the virion RNA, the negative strand, into messenger RNA. So messenger RNA and virion RNA for VSV were opposite strands. And it implied an RNA polymerase in the virion, and in fact, it was very obvious once we assayed for it. And we found many other lipid-containing viruses that had negative strands, and realized that there were two broad classes of RNA viruses, positive strands and negative strands. And so that then enabled us to make this little diagram of how viruses make their messenger RNAs. Some DNA viruses, of course, either through a single strand or double strand of DNA, make messenger RNA. But RNA viruses can either be positive strand or negative strand. And the positive strand viruses make a negative strand, which then is the template for more messenger RNA, whereas the negative strand viruses directly make a copy of them, of the long RNA into a subviral messenger RNAs. And there are double strand viruses. So we had a very compact view of what was going on in virology, except that there was one class of viruses that didn't fit this picture. And they were the RNA tumor viruses. And they were viruses that were known for their biology, not for their structure. And their biology was that they could cause tumors in animals. And of course, Howard Temin, who has already been introduced, and Rubin had figured out how to quantitate the tumors that were made by RNA tumor viruses and turned cancer research into quantitative virology. So we had the genetic strategy for almost all viruses, but Howard Temin had the insight 10 years previously. We're now actually in 1970, but 10 years previously, when he was at Caltech, he had the understanding that RNA might be copied into DNA. This violated the central dogma of biology and was treated largely with scorn by the rest of the scientific community. And in spite of it, Howard for 10 years had been looking for an experiment that would convince somebody aside from him that this was possible. And I saw that the way I had been doing science up to then, looking for polymerization enzymes, was the right way to look at this problem. But the problem was finding a source of enough virus to have a chance of seeing an enzyme. And luckily, the NIH had started the Special Virus Cancer Program, a program which was designed to ultimately show that viruses caused human cancer. And that was, in fact, not possible because the RNA tumor viruses cause very little human cancer. But they were stockpiling virus in case somebody one day had an idea for how to study these viruses. So I called up the people running this program and said, can I get some virus? And they were very excited that somebody cared and sent me a lot of virus. And I started analyzing the virus for a DNA polymerase. And this is the page from my notebook of the very first experiment that I did. And for any of you who've done biochemistry and are in the sort of school of Arthur Kornberg, you'll recognize this format as the format that disciples of Arthur use. And I'm one of them, having worked with Jerry Hurwitz, who was a student of Kornberg's. And it involves making a mix of things that every tube is going to have in the assay, with buffer and magnesium and a sulfhydryl reagent, and DATP, DTTP, no DCTP or DGTP. And then this mix is distributed among, in this case, 9 tubes. And in many of the tubes, I added back DCTP and DGTP. So if there was a polymerization enzyme it would have all 4 nucleotides to polymerize. And then there were a couple of tubes that lacked DCTP, DGTP. They should have no enzyme activity at all because they can't copy C and G. And I incubated some of the tubes, and some of the tubes I kept cold, and got various counts because there was radioactive TTP in there. Got various counts, and I won't take you through all of them, but they effectively are random numbers. And I should have maybe given up at that point. But I looked at the numbers. Some of the numbers were large. Some of the numbers were small. I said, maybe there's something there that hidden away. So I made this note to myself, might have something there. Alice, my wife, has always said that that's one of the most optimistic notes she's ever seen. And now, 2 days later, I came back to continue the work. I don't know what I did in that day in between. But now what I did was to take some of the virus that had been sent to me from NIH and spin it down in a centrifuge into a pellet. So it was very concentrated, taking it up in a minimal amount of buffer. And now put that into a reaction, again with DCTP, DGTP, and the various bits of the mix. And now I got a remarkable result, because the unincubated tube, there's only 4 tubes in this experiment, had 100 counts per minute. The incubated tube with a certain amount of virus had 650 counts. With twice as much virus, it had 1,300 counts, basically twice as much activity. And without DCTP and DGTP, there was nothing significantly over background. And then I concluded that there is an enzyme, a DNA polymerase, in the particles of RNA tumor viruses. And that was the discovery of the reverse transcriptase. Basically, a couple of days' work doing things that I had been doing for my whole career. Now, what I'd shown you doesn't say that there was an RNA template, but we could quickly show that there was an RNA template because their reaction was ribonuclease sensitive. And soon thereafter, Inder Verma, who was a new postdoc in my lab, purified the enzyme and showed that it would accept any RNA as a template if you provided a DNA primer. Now, we called the enzyme the RNA-dependent DNA polymerase, but Nature magazine, being a journalistic enterprise, renamed it reverse transcriptase because it reversed the standardly accepted direction of information transfer in biology. And the fact that it would copy any RNA with an appropriate primer really was the beginnings of biotechnology, as was said earlier. So it was effectively 1 week of experimentation. There was a week hiatus in the middle of it because everybody I knew went out on strike, including me. in that week to protest the Vietnam War. And it's an interesting parallel to life today. So there were many consequences of the discovery of reverse transcriptase. First of all, it filled out the understanding of viral genetic systems. And so now we could add to the picture that I showed you before a class of viruses that have RNA as their genetic material, but copy that into DNA, and the DNA then is a template to make messenger RNA. And this compact little diagram describes how all viruses, the life cycle of all viruses, and you can actually separate the viruses into classes this way. And this became the Baltimore classification, which many of you may have been subjected to. I never expected it to be named for me, and people still think it comes from the city of Baltimore. The second consequence was that the central dogma could be reversed. And that's actually a very general statement that RNA can be copied into DNA, and that the what was then thought of as a very transient molecule, RNA, the information from it can be stored in DNA. And so the central dogma of molecular biology, which at the time was the DNA is replicated, it can be copied into RNA, and the RNA then can be copied into protein, became an updated dogma in which reverse transcription allows RNA to be copied into DNA. A third consequence was not a logical consequence, it was a general understanding that cancer could be a genetic disease because the virus that caused cancer, the Rouse sarcoma virus in particular, made a copy of itself into DNA. And we could easily imagine that that DNA goes into the nucleus of the cell and is integrated into the cellular DNA. And although we didn't show that, it took a number of years before that was shown, it meant that cancer was likely a genetic disease. And that became the direction of most cancer research ever after. A fourth conclusion was, as we've mentioned, that you could capture messenger RNAs as cDNAs, and that became the beginnings of the biotechnology revolution, which we now bring up to date with CRISPR, so that you can make targeted changes in inheritance. The fifth conclusion was that large parts of the genomes of organisms do arise from parasitic DNAs, that effectively what viruses are, the reverse transcription containing viruses, are parasites of the cellular DNA. A sixth conclusion took 10 years before it became obvious, and that is that there's a virus that causes the disease of AIDS. That was in 1980. And I've always thought that it was a good thing that we had found the virus, we had found the reverse transcriptase 10 years earlier rather than 10 years later. And finally, it led to my winning a Nobel Prize, along with Howard Timmon and Renato Giabecco. And as was pointed out, I was 37 years of age. I didn't think I was so young, but looking back on it, I certainly was that young. Let me tell you how I found out that I'd won the Nobel Prize, because it's an amusing little piece of history. My wife, Alice Wong, who is, as we've said, a virologist, was at a meeting in Scandinavia. I was home with our one-year-old baby, and I got a call early in the morning from her. What had happened was that she had given a talk at the meeting she was at, and that at the end of her talk, George Klein, who was a great Swedish virologist, had done what he did so often at meetings, which was to summarize the sessions that had taken place, and the talks that had been given. And he did this, as Alice has said, sort of endlessly. And he finally said, I'm trying to use up time here, but I can't. And I'm going to tell you who's going to win the Nobel Prize, which will be announced in half an hour in Stockholm. And he announced that Howard Timmon and Renato and I were to get the prize that year. And Alice ran to the telephone and called me, woke me from a sound sleep, and told me about the prize. It was a unique way of learning about a Nobel Prize, one that I've never heard anyone else come close to. So let me thank you very much for your attention. If there are any questions, I'll be very glad to take them.
I don't know if this is on.
Can you hear us,
can't.
Professor Baltimore?
Now I can, yes.
Fantastic lecture. This is Ruben Harris from UT Health. Do you happen to recall the name of the Nature editor that was so bold to change the name of your enzyme?
I think it was the editor of Nature, and I'm not going to come up with his name, although I'm sure there are people in the audience there who know him.
David, it's John Coffin. It was John Tooze.
John Tooze, oh.
And who actually confessed it to me at a meeting shortly afterwards that I happened to be sitting next to him at.
Aha. I thought it was the editor in any case,
He was a section editor, I think, at the time, actually.
journalists have a different view of names for scientific materials.
So thank you for this beautiful talk. So I was wondering, when you got the Nobel Prize at 37, how do you feel? Aren't you afraid for the rest of your professional life? What are you going to do after that?
No, I got into science because I love data. And the only thing I really care about in life is data coming from my lab, data coming from other people's labs, data coming from my own hands, although that stopped many years ago. And for that reason, I didn't stop doing science because I'd been given an award. I thanked the people who gave me the award. I appreciated it very much, but I went on to continue in the science. In fact, I took the opportunity to switch a lot of my work from virology to immunology, because I saw that there was tremendous opportunity in immunology to apply the then just developing methods of recombinant DNA to the immune system, in particular to the B and T cells, to understand how they develop their specificity. And we were able to find the enzymes that did that. It's always, for me, looking for enzymes. And that kept me in data for many years thereafter.
David, this is Ron Swanstrom. I'm
Hi.
Pharmacy Bishop trainee along with your host, Steve Hughes.
Right.
I have a confession to make. There
Yes,
was
sir.
a time when you went off to do a sabbatical, I believe it was in Leonard Erickson's lab, to try to figure out how to make Paul after YAG, and worked out the suppression of the tRNA, the termination site with the suppressor tRNA, and you came back to Cold Spring Harbor to give us that talk. And it was the same year that here at Cold Spring Harbor, they demonstrated splicing. And I just thought, oh, David's got it wrong. It's going to be splicing. And, and of course, I was a fool's errand to second guess you, so I apologize.
Well, I was even involved in the splicing discovery. We're advising Phil Sharp.
This is Paul Beanash from Rockefeller University. So, for those of us that have grown up not knowing a world before reverse transcriptase was discovered, I'm curious to know, given all that was known about the biology of RNA tumor viruses, heritability, it seems to us, looking back, that it was sort of obvious that reverse transcriptase should have existed. But when you actually did the experiment, were you already convinced that the enzyme existed and all you needed to do was to design the experiment that would show it would exist? Or was there genuine uncertainty about whether the enzyme actually existed? And sort of a related question, when you got that result, which is, it's one experiment, how sure were you that you were correct, right?
Well, let me go back to the first question. Was I convinced that it would be there? No, I wasn't. In fact, when I was searching for virus to do the experiments, I had received from one person a small amount of Rous sarcoma virus. And the first experiments that I did, I looked for an RNA-dependent RNA polymerase in that material because that seemed like the simplest explanation, which was that this was another lipid-containing virus that worked the same way that VSV did. But I got a totally negative answer from that experiment. And I said, well, either there's a DNA polymerase here or there's nothing here. And then arrived the virus from NIH. And I did the experiments which I showed you. And that 4-tube experiment that I showed you did convince me that there was an enzyme because I had been doing that kind of experiment for so long that I knew the kinds of controls to build in to be sure that there was a temperature-dependent synthesis of DNA going on in that test tube. And so I had no more question in my mind, but that Howard had been right for all of those years.
Tom Hope from Northwestern University, and I guess I'm wondering where you would think the most interesting data is coming from in the future to help all the young people here that are trying to find careers coming up.
Well, unfortunately, the place where exciting data is coming from these days is from AI. And I say unfortunately because AI isn't a logical formulation. It's a way of looking at history. And it is a fabulously insightful way of investigating history. But it doesn't tell you how it gets an answer. It just gives you the answer, which isn't satisfying at all. So I think coming to terms with the power of AI is the forefront of science today, of our science today.
Richard Sutton, Yale. A related question. What is the future of science in the U.S.?
Dubious, unfortunately. With the federal government cutting down its support of science so dramatically, I think we run the real risk of falling behind the rest of the world, particularly the Chinese, and becoming a second-rate power in science. I'll be looking very carefully at what happens over the next 4 years. In fact, maybe the next 2 years, to see if there's a chance of keeping enough of basic science intact that we can start it up again in a different political framework.
I guess a related question is, what can we do sitting here now?
Not a whole hell of a lot. I think what you're doing, which is to pretend it's ordinary science, and to do what's been done at Cold Spring Harbor for so many years, is the right thing to do. But I think we also have to be cognizant that we may be the last generation of Americans that have the opportunity to be talking about basic scientific developments.
Professor Baltimore, thank you so much for your insightful talk. It was very exciting. I'm Ganjam Kalpana, one of the trainees of Steve Goff. And my question is, so according to you, what is the one important thing that we still have not understood about the RNA viruses?
Oh. You know, one of the things to think about is the small peptides that are made by many different viruses, maybe all viruses, and that have important roles in the life cycle of the virus, but that we haven't yet found exactly what they do and why they do it. So I would keep an eye on those.
Interesting.
The other thing is to keep an important eye on new viruses that are emerging. Because every virus brings with it open reading frames that don't look anything like what we've seen before. And those are opportunities for discovery, often of brand new ways of thinking about molecular gyrations.
Thank you.
Is there anyone else who has a question for David Baltimore? If not, we should all say thank you very much. That was lovely.
Thank you for your attention.
All the very best. Thank you.
Thanks for your remarkable introduction.
It doesn't do you justice.